Good morning, everyone.
Morning.
It's a pleasure to see all of you already connected. So today we have the talk of Professor Martin Larsen. Before introducing him, I would like just to remember you that the certificate for attending this lecture will be actually the link for the certificates will be sent at the end of the talk, and also the link for sending questions through WhatsApp will be just sent now as soon as we start. So it's a pleasure to introduce Martin. So Martin is currently a professor, a full professor at the University of Southern Denmark. He obtained his PhD in proteomics and biological mass spectrometry in 2000. And since then, he had a wonderful career, being associate professor in 2009, and then full professor in 2011 in clinical proteomics. And he's also the head of the Center for Clinical Proteomics in Odense at the University of Southern Denmark. So his publication records are really impressive. So he has almost 200 papers. And he has been actually mentoring several PhD and master students, also supervising several postdocs. And along his career, actually, he received several prizes. One of them, I will just remember one of them, the most recent one, is the recipient of the Danish Elite Research Award, which is actually a very important award that is given directly by the Queen to the researchers that have been actually distinguishing their career in science. So Martin is very well known for the development of methods that have helped many scientists to study signaling pathways, especially through phosphorylation. But he also has developed several methods in other PTMs. And today we'll talk about the enrichment of PTMs. He called the version 2.0 because he's always working in the lab developing new methods for us to use and to apply to our system. So, Martin, it's really a great pleasure to have you here. I will just remember that we are on the online seminar, My Friend is MASSA, which is supported by the BrPROT. So, and it's a great pleasure to have you here. And I'm sure everyone will have a lot of questions at the end of your talk. Thanks, Martin.
Thank you very much for the introduction. And it's always nice to be back in Brazil, or at least virtually. Right now, I'm at 10 degrees in Denmark. So my title for the talk today will be Enrichment of PTMs, version 2.0, as Giuseppe said. And I will try to give you an introduction to PTMs and what we are doing in the lab. So first of all, the overview of my talk will be a short introduction to PTMs. Then I will go more in detail into phosphoproteomics, into sialylation, which is a special part of glycosylation. And then I will give you a few examples of how to use some of these methods that we have developed over time. And at the end, I will end with some thought about cysteines and modification of cysteines and how to look at cysteine modifications in general. As you probably all know, proteomics is a bit more complex than we thought maybe 20, 40 years ago. We have made a big deal into genomics, where we have found out that there are probably more close to 20,000 different genes in our genome. But then you can modify this so that you have actually the outcome will be higher. So there are alternative splicing and the RNA editing. So we have many, many more transcripts. So the transcriptome is much higher. But then nature have kind of been playing around with us proteomics people for many years. And it turns out that many of the proteins we have in our cells are enzymes that are capable of putting on different kind of small molecule onto the proteins, the other proteins, so that you can change their function or activity or any other things with the proteins. And actually, between 3 and 5% of all proteins that are present in a cell is an enzyme that are participating in these post-translational modifications. And that means that going from maybe 100,000 possible proteins, we can now make millions of different protein molecules in a cell. And each of these protein molecules could potentially have a new function or new interaction or any other things. So it's a bit more complex than looking into the genome when we do proteomics, and especially when we do the proteoforms or the post-translational modifications. So there are many examples of PTMs. So here's the phosphor, glycosylation, lipids, but there are actually quite a lot of different PTMs. The function of PTMs are very diverse. So you can have some of the well-known for many years modifications is the signal peptides, which is sitting in front of proteins, or which is synthesized first in proteins that are going to be synthesized into the ER, and then further on out to the plasma membrane or any other organelles or secreted from the protein from the cell. And then you also have molecular switches, and that can go on and off. You have a lot to do with cell signaling. Stability, solubility, correct protein folding are some of the things that, for example, rely a lot on the glycosylation. And we have many other functions of PTMs. And one of the things why we should study the PTMs in proteins is, first of all, we want to study the dynamic in biological systems, but we also have found out in many cases that not only in the biology, but also between different diseases or the initiation of diseases, we don't see a change a lot in expression of proteins, but we can see many times that it's exclusively on the post-translational modification level. So this means that if you're just using normal conventional omics methods, you will never have a chance of finding out what is actually behind these diseases. As I said, there are many modifications, and up till now, I think there are about 400 known chemical modifications. And there's also quite a lot of these modifications that we introduce ourselves during the sample preparation. So you have to be careful when you go into modifications. So oxidation of methionines and also oxidation of cysteines and other amino acids, you can introduce yourself when you're doing a sample preparation. One of the bad things within PTM analysis is that there's not that many strategies or strategies for characterization of these modifications. So actually, we only have enrichment strategies for a handful of these modifications, and these include, for example, glycosylation or N-linked and O-linked. The maybe 10 different modifications you can put on lysines, like acetylation and methylation. This is mainly based on antibodies. And then you have, for example, phosphorylation, which is one of the most abundant modifications that people have been studying the last 20, 30 years using mass spectrometry. And all these different kind of modifications, we have actually quite nice methods to enrich for them, and that means we have a possibility for study these ones. When you look into the workflow of proteomics, you will see this is the very conventional workflow. I think most people that are doing proteomics are familiar with this, that you have to come up with a sample preparation stage 1, where you extract your proteins from your either tissue or cell cultures. And this can be done in many different ways. Common for all of them is that you proteolyze your proteins into small pieces, peptides. You can also go the other way and then do top-down proteomics, but there are not many people in the field that are doing this still. Most people, they digest into peptides, and then you can put these peptides onto your very new and advanced mass spectrometer that is shown down here. We are actually using some that are a little bit more novel. And then you do data analysis, get a lot of identifications out and a long list of proteins or peptides. And if you do this, it's very rare that you see many modifications. For this, you need to enrich them. There are some publications out where they claim they have found modifications, and you can find modifications if you just do the normal data analysis down here. But if you enrich for the modified peptides and you have a good enrichment method, then you can see many, many more peptides that are modified. And one of the reasons for this is that when you do the normal analysis, you rely on LCMS, for example. So the LCMS, you have reverse phase chromatography, and many of these modified peptides, they're hydrophilic, so they can risk to go right through your column. They have low stoichiometry, so maybe only 1% are modified of this given site at the same time. So you don't see these things. Some of them can also be labile, so they actually fall off during the sample preparation. So enrichment of modified peptides is essential if you want to be good in PTM analysis. If we look into the various PTMs, then of course protein phosphorylation is a key modification. And this is a modification that most people can relate to. And most people have been struggling with doing phosphoproteomics analysis or phosphorylation analysis the last 50 years. Protein phosphorylation is participating in almost every single regulation process in the cell. So we see here surface receptors are driven by phosphorylation. When you divide cells, it's all a phosphorylation driven process. So many of these fast processes, also contraction of muscles, they don't rely on maybe making new protein, but they rely a lot on, is the phosphorylation increasing on this specific amino acid in a protein? So what is this phosphorylation actually doing? So if we look at the common way to look at protein phosphorylation, we have kinases and we have phosphatases. And we have proteins that are getting phosphorylated by the kinases, and then the phosphatases are taking off these phosphorylations again. And one thing you should know if you want to go into this field with phosphoproteomics is that phosphatases are 100 times faster than kinases. Which means that when you do your sample preparation and you want to look at short time stimulation, for example, you really need to kill the phosphatases. Otherwise, you get a mixture in your sample. There are around 537 kinases, and there are almost 200 protein phosphatases. There's also lipid phosphatases and other phosphatases. So now we have put on a phosphate group on this given protein here. So what can we actually do with this phosphate group? So many people, when you read the literature, they say that when you put on a phosphate group, it becomes activated, or it can also become inactivated. But probably most of the phosphosites that are on proteins are not doing any of these two things. But most of the bioinformatics that you look into are actually relying on the fact that it is either activating or inactivating. So what we think, and what we have also shown earlier, is that when you put on a phosphate group onto a protein, the local environment is changing. And this local environment is very, very important for protein-protein interaction. So many of these phosphorylation sites that are put on are actually put on to generate new protein interaction pathways. So when you have done this interaction, then you have an effect. So this is probably the most general thing to do, and also we have the activation, deactivation. So activation, deactivation, and interaction are what protein phosphorylation is doing. But it's very important to know here that not many phosphorylation sites people know what to do, what are doing. So it takes years for researchers to find out what a specific phosphorylation site is doing. So many of the phosphoproteomics papers that you will read, they are assuming, and they use pathway analysis programs to assume a lot of things, whereas you probably should go and validate many of them. When you talk about phosphorylation, you talk always about cellular signaling and signal transduction. So it's very important. So phosphorylation is very important in cellular signaling, and you can see here. So this is a plasma membrane, and in the plasma membrane, you have a lot of receptors, and these receptors are measuring what is outside the cell. So if you have a growth hormone, for example, coming to approach the cell, there will be a receptor for this growth hormone, and when this fits together, you will start a phosphorylation cascade into the cell, and that will ultimately end up in an output, and most of the cases the output is synthesis of new protein inside the nucleus. But there are also many other things that happen. Common for this is that this is driven by a cascade of kinases. So you can activate a kinase that can then activate another kinase and a fourth kinase and so forth into the nucleus and other places like the mitochondria, for example. So one thing to have in mind when you're looking at the phosphorylation and you want to characterize signaling pathway is that sometimes it's easier to characterize the distal kinase profile. So when you have the signaling, you start out here, for example, out at the plasma membrane, you have kinase 1. And this kinase 1 can be activated, and then it will activate maybe, for example, 4 molecules of kinase 2. And these will each activate 4 molecules of kinase 3, for example. And then you will have this amplification that get higher and higher the further you get away from the plasma membrane and into the cell. And this has actually some complications when you want to look at dynamic of phosphorylation. First of all, it's sometimes very, very difficult to find out the phosphorylation sites on the kinases that are out next to the plasma membrane, because you will have a much, much lower stoichiometry of this. So maybe only 1 out of 100 kinases, same kinase 1 here, will be phosphorylated and activated. Then you'll have a little bit more of the kinase 2 and more of kinase 3 and much more of kinase 7 that are very close to the center of the cell or inside the cell. So one thing you should, what you should consider for phosphoproteomics analysis is the speed of the signaling, and especially here, how to stop it again. So you want to stop it at specific points, but it's not all reagents that stop signaling, and spinning down or centrifugation does not stop signaling. So you need to stop the phosphatases also in this game, and you need to look at the abundance of the phosphorylation. Are you really in a position to find out what is out next to the plasma membrane? And this has to do with stoichiometry. So there's a lot of things to think about when you want to do the phosphoproteomics setup here. So before phosphoproteomics was developed, many people were looking at protein phosphorylation and signaling pathways using antibodies. And there are many antibodies that have been developed to specific phosphorylation sites over the years. So for example, here in this interleukin-1 beta signaling in beta cell, pancreatic beta cells that secrete insulin, you have this interleukin-1 beta receptor. And if you stimulate beta cells here, this receptor will start and you will generate these receptor complexes. And that will kind of fire this signal transduction into the cell. And then you will go through the ERK-MAP kinase pathway and the NF-kappa beta pathway, and that will lead to expression of proteins. Unfortunately, in beta cell, one of these proteins is the eNOS protein, and that will lead to production of NO, free radicals. And if you have a high amount of free radicals in beta cells, then you will start these apoptotic pathways. So this is not a good way to do it. So beforehand, people have been looking at these protein phosphorylation, and especially the ERK-MAP kinase is very, very famous, the antibody against these phosphorylation. But the problem is that you don't look for other things. So you're very biased in your analysis because you only look for this particular protein phosphorylation. So by doing phosphoproteomics, we cleave things in pieces and then we purify the phosphopeptides. And that means that we can actually look at many, many more things in one go. And if we combine it with some quantitative studies like multiplexing, then we can see many different conditions at the same time. And it's not uncommon. So right now, as Giuseppe said, I am still in the lab. And it's not uncommon for us to get 20,000 unique phosphorylation sites in one go with the methods we have right now. So we see many new things that people have not been looking at before. And one of the things we see with interleukin-1 beta signaling is activation of proteins that are sitting in the mitochondria also, for example. And this you will never see with any of these antibodies. So when we want to study protein phosphorylation, you need to have a good way to enrich the phosphopeptides. And for many years, people were playing around with IMAC, which is a method that rely on an iron or gallium complex that make a complex with a phosphate group, but it also has a high affinity towards these negative charged amino acids. So for example, down here, if you only use acetic acid in the enrichment, you see a lot of non-modified or non-phosphorylated peptides that you drag out of a solution. You can increase the amount of phosphopeptides in this by putting the peptide solution in 0.1% TFA, which is a bit stronger than acetic acid. But the moment that you go up to a very complex mixture, we and many other people have very big problems with IMAC. There are some publications out there, novel publications, that show that you can, if you set up IMAC in a column form, that you can get quite nice enrichment. We have been developing a lot on the titanium dioxide chromatography that I will go into detail a little bit later here. And then we have the phosphotyrosine antibody pull down, and this is actually only doable with for the phosphotyrosine. So we have the tyrosine and serine and threonine. The serine and threonine antibody does not really work. It's basically only the phosphotyrosine that work for pull down strategies. And I think it's because that the two other amino acids, they're too small, so they don't generate a good and specific epitope. You can also, if we go back to here, so you can also get antibodies that are recognizing a specific motif. So for example, protein kinase A motif, you can get antibodies that recognize this, and some people have used them with quite high success in phosphoproteomics. So if we go back to titanium dioxide, it was developed in 2004 by these three groups. And all of the groups used the same conditions. So they loaded in acetic acid, and they could purify or enrich for phosphopeptides. But in all of them, you saw that you had a very high amount of non-specific binding. So if you go on a higher complex sample, people in these studies here suggested that you did O-methyl esterification to block all the acidic residues in peptides. And I cannot recommend this unless you have a very, very nice method, because most of them will give incomplete methylation, or it will actually deamidate places that where you then will put in methyl groups. So we played around with this in 2005, and we came up with this method that we published in MCP, and it's a very strange method. So our protocol here is basically that you load in this very weird solution with extremely high amount of TFA, and then we add this glycolic acid. So in the beginning, we added other acids. So we had DHB that are used for moldy. That's where what we developed it on. But then we saw a lot of contaminations when we did the DHB enrichment with LCMS. So we turned to other acids, and then we came up with this 1 molar glycolic acid that seems to work quite nice. Then we wash with these solutions here, and then we elute with high pH. And this is just an example with alpha casein. So this is loading in DHB. And if you do a moldy dry droplet method here, we can see 3 phosphopeptides. If we load onto a titanium column in DHB and elute, we get a lot of phosphopeptides and even some contaminants that were not seen before here. So this is a method that works quite specific, and I think most people that have worked with it over the years since we developed this see more than 80% pure phosphopeptides, even for people that have not really been in the field and have just started out doing it. So it seems to be a quite nice and reproducible method. Why are we adding glycolic acid or DHB? We don't really know. So it works. We think that this is because that acidic amino acids, so the acidic group can bind in the same way as DHB or glycolic acid, whereas the phosphate group can bind in this way here. And this binding here is much, much, much stronger than this one over here. So by adding an acid that is multifunctional, you can actually exclude many of the acidic binding peptides in this case here. So now we have methods for purifying phosphopeptides. So there is titanium dioxide that we are using a lot. Then there's the IMAC that many other people are using. So now we want to set up a large-scale proteomics study. So how are we doing this? Because we cannot just purify phosphopeptides and then run them directly, because then we only get a limited amount of identifications out. So many people, they want to do a fractionation, and what kind of fractionation should you do, and when should you do it? So many people are playing around with pre-fractionation, and here you take the peptides and purify by any means here into maybe 40 different fractions or 20. And then each of the fractions they purify by IMAC or titanium dioxide or tyrosine phosphorylation or pull down. And then they run each sample on LCMS. And this is to reduce complexity prior to the phosphor enrichment. And this is really, really important for IMAC. And it's not so important for titanium dioxide to reduce the complexity, at least not in our hands. You can also go the other way around. You can do the enrichment first, and then you can do the separation afterwards. There's a big advantage in this, is that first of all, you're not handling 20 or 40 enrichments up here. You only handle 1 or 2 enrichment, and then you elute phosphopeptides, and then you either run high pH or helic, and then you get it out in maybe 30, 20 or 40 fractions here. And then you can dry it down and put it directly in LCMS. If you chose to do the post enrichment here, you can either do helic or this high pH reverse phase. You want to choose an enrichment method, or not an enrichment method, but a separation method that is orthogonal to reverse phase LCMS. And there, these two have the highest orthogonality. Many people have used strong cation exchange, and many people are still using strong cation exchange, but they don't have the high orthogonality here. And then you can think about which one of these two are the best one to use, and which one should you use. So if you look into phosphopeptides, they are in general hydrophilic of nature. So it also depends on what kind of quantitative analysis you want to do. And it depends on if you want to do the fractionation post or pre-phosphopeptide enrichment. And one of the things that we have seen here, so this is helic separation of phosphopeptides. So we see a nice separation here. And then we have the high pH reverse phase separation here. With some of the, at least in our hands, the high pH is very nice, especially if you have a nice fraction collector that can allow for concatenative collection, so that you maybe collect in 20 fractions, but you have a collection every second minute for more than 2 hours. And that means that fraction 1 and 21 goes into the same vial, and so forth. That can have a lot of advantages, so that you use your fast mass spectrometers you have these days. I mentioned TMT here, and there's a good reason for this, because TMT and iTrack are actually adding a hydrophobic group to your peptide. That means that if you TMT tag your phosphopeptides, then they will become more hydrophobic. And that means that if you put them on helic, they will elute before, and they will elute in a much sharper peak, so you get less fractions. If you have naked phosphopeptides, they will elute in a very nice separation like this, and you get more fractions. If you go to the high pH and you have a naked phosphopeptide, it will many times come out in the flow through. But if you tag it with TMT, it has a higher hydrophobicity and stick to the column, and then you get a nice separation like shown here. So it depends on what you want to do, what kind of fractionation you should use. Also, if you go into the literature and you try to compare what other people have been doing, there are a lot of things that you need to think about when you read these papers. It's never the same MS and LC instruments that are used, and this gives a high bias to the people that are using it. So it's very difficult to compare between methods. It's never the same sample, so you compare apples with bananas. It's never exactly the same protocol that are used in all these published protocols. It's never the same peptide identification software and identification criteria used. And then one person cannot be expert in all methods. That means that you have a bias towards the method that people are developing. So if someone develop a new method, do you think that this person will be biased towards the method in a direct comparison? I strongly believe so, because if the method that you are developing is not better than the current methods, you have very big difficulties in publishing. So this was about phosphorylation. So now we have means to purify phosphopeptides. We can apply it to large-scale phosphorylation, and we also have quantitative means like TMT or Silac or other things, so we can compare different samples with each other. And I will come back into this when I show the examples. One of the other PTMs that we have been looking at for quite some years now is glycosylation. And we're looking at, so all know that glycosylation is made in the ER and Golgi. So when the proteins are synthesized into the ER, there's a lot of enzymes that first put on a big glycan structure, and then they cut it into pieces, and then you build it up with the use of a lot of different enzymes into the ER, and then they are transported to Golgi. And in the Golgi, there are a lot of enzymes for putting on a special sugar unit called sialic acid. Sialic acid has this structure, at least in human. And you can see this is a 9-carbon sugar unit, and it's sitting out here. So if you have N-linked glycosylation that sits on asparagine here, we can see this is the N-linked structure. And then out here at the far end, we see the sialic acids. And sialic acids are quite negatively charged. And these are actually the first thing that anything that approaches this cell will see when they come closer to it. So basically on the surface of cells, we have this negatively charged sialic acid sitting out. And it's quite interesting, sialic acid. There's a lot of viruses that bind to sialic acids. There's a lot of viruses that has a lot of sialic acid on. There's a lot of pathogens that are binding to these sialic acids. So some years ago, actually in 2007, we wanted to do the plasma phosphoproteome, and we found that there's a lot of things that bind to titanium dioxide in plasma. And the most abundant modification in plasma is actually glycosylation with sialic acid on. So we found out that we can enrich for sialylated glycopeptides very, very nicely with titanium dioxide. So we came up with the use of the sialiome, not to mention the psyllium, which is the saliva proteome. So, and we have been using titanium dioxide for many years now for studying sialic acids. And recently here it becomes more and more interesting modification. So for example, beside being involved in a lot of things for migration of cells and cancer and all this, we have been very interested in this evolution here. So as you can see out here, we have the human up here, and we have chimpanzees, and then we have the other great apes here. So at some point we were kind of sticking out from the chimpanzee, and this means that we are the closest relative we have in the animal world is the chimpanzee. And we only have 1.5% different genome between us and the chimpanzee. And there are a lot of people that are looking at proteins between us and the chimpanzee, and there are not many changes. One of the biggest changes is actually sialic acid. So in the sialic acid biology, there are around 60 enzymes that are involved. And out of these 60 enzymes, 10% are changed between us and the chimpanzee. So this is a huge difference compared to the proteome or genome. So in our cells, we have the human version of sialic acid, which is this one. And in most primates and other animals, they have this one and this one. So they have a combination of both of them. And one of the reasons that we don't have this this one that they have in primates is that we lack the enzyme for making this. And then you can of course say that, okay, we have the enzyme, but it doesn't work. But maybe some people has it. So maybe there are some traces in some humans. As we can see here. And if you go further back in evolution, there will also be at least something pointing in the direction of evolution taking place in America, I guess. But the sialic acid binds really well to titanium dioxide. So maybe we can study this evolution in the future. So this is actually one of the things that we want to do in my group here in the future. So because it binds so well to titanium dioxide, we have come up with a further development of our comprehensive workflow for phosphorylation. And what we are doing, since both of them are binding to titanium dioxide, we can enrich for both of them in a sample. And that means we have a mixture of phosphopeptides and sialylated glycopeptides. To look at intact sialylated glycopeptides, it's very, very difficult. And it's even more difficult to identify intact sialylated glycopeptides compared to neutral glycopeptides. But I won't go into detail with this. So what we are doing here, we are deglycosylating, and then we have a handle because we turn the N to a D up here when we cleave with PNGSF. That means that if we then do a second round of titanium dioxide, you will have the phosphopeptides will become bound to the second round here, and then the flow through will not have sialic acid on, but will be naked peptides that has the link for the marker for the deamidation. And that means that you can actually separate these two species from each other. And this we published first time here in MCP in 2010. So we have been using these methods, or at least this that are shown before here. So this method here, this workflow, we have used many, many times after 2010. And I will show you why it's good to look at both sialylated glycopeptides and also phosphopeptides in the same sample. And I will show this by looking at glucose-stimulated insulin secretion. So we had a paper out in 2018 from Taiwo Kang and Pia from my group. And they have been looking at pancreatic beta cells. So they purified pancreatic beta cells from small rat pups. They actually used, for this study here, they used 180 rat pups. And then you purify the isolate of Langerhans, and then you let them grow for a while, and then you can look at the stimulation of the beta cells. And the way you stimulate the beta cells is by adding glucose. So when glucose come in to the glucose transporter, it's degraded and you generate ATP. So when this ratio ATP-ADP goes up, then the ATP will block this potassium channel, and this will result in a depolarization of the plasma membrane. When this depolarization is taking place, calcium molecules will come in, and the calcium will initiate calcium signaling inside the cell, and this will initiate exocytosis of insulin, but also endocytosis of plasma membrane out here. So insulin is stored in these granules inside the beta cell, and they are released because of this calcium influx. So what you see if you measure the insulin from beta cell is that after stimulation, when you add more glucose, you have a very high peak of the initial insulin release, and then it goes down and becomes stable up to 40 minutes or so. When you then remove the glucose again, it takes maybe 5 to 10 minutes, and then it goes down to normal. So this is also what happens in your body, for example. So we wanted to study this phenomenon. So we wanted to take several time points over the first initial starting point here. So we tried to do this with isolating the isolate of Langerhans, stimulating for 5, 10, and 15 minutes. And that will cover the initial release and also the prolonged release. So if we look at, so we did non-modified peptides, we did phosphorylation and sialylation, and you can see here that we have a quite nice coverage. This is some years back, so we didn't have the very fast instruments for this study. But when you then start doing some pathway analysis, you can see that based on the phosphorylation, we can see that there's a huge increase in many different signaling pathways inside the beta cells when you stimulate with glucose. And many of them make completely sense. Many of them are actually because there's many of the proteins are shared between different pathways, and that means that some strange pathways comes up when you do this. But we have the calcium signaling in here. We have a lot of other different kind of signaling. One of the signaling pathways that we were very interested in was the integrin signaling. So if you look at integrins, these are surface proteins that are linking up to the extracellular matrix. And integrin is known to link to the focal adhesion kinase via a lot of different proteins. So we did this for 5, 10, and 15 minutes, and then we did the phosphorylation and the glycosylation. We didn't see much changes in the non-modified because we don't expect to have any increase or decrease in proteins within 15 minutes. But if you look at the sialylation and the proteins that are regulated by sialylation in this phenomenon here, then already after 5 minutes, we see a regulation in sialic acids. And this is not enough to say that these proteins here, they are changing in sialic acid because of their abundance. These are changes in sialylation on the surface of the cells. Because on the surface of cells, you have neuraminidase that can cut sialic acid down. And you also most likely have sialyltransferases that can put on sialic acid, but this is still speculated. But you can see if you take all the changes and regulation in sialylation, we see a lot of extracellular matrix proteins, also the integrins, and then we see this semaphorin plexin interaction and other things taking place in organelles here. So if we then take these two results and put them together and look at integrin signaling, then we can see that there is a very nice link here. So here we have focal adhesion because we have the integrins up here that change in sialylation. So one of the things that integrins are communicating with is sialic acid. So integrins link up to many of its ligands via the sialic acid, and it's actually a very important molecule for cell migration. So this indicates a strong or a crosstalk between sialylation and phosphorylation inside the cell. So if we look at, these are the integrins, these are the complexes that are formed here. There's the focal adhesion kinase and there's the paxillin. So we saw the paxillin signaling pathway, and we can see that this leads to actin organization. So this is the focal adhesion, and then it leads to a lot of different things in the cell. And if you then remember that I also talked about the semaphorin. So the semaphorin is a bit similar. So they are out here, and they also leads to changes in the phosphorylation inside when they get activated, and they are also regulated on the sialylation level. So this here, you can see the integrin link up to the extracellular matrix, and they are linking up via the sialylation. And you can see all the pathways that you are linking to. And these are the pathways, the same pathway we see as upregulated when we stimulate with glucose. There's also a paper here that supports this, because what we see here is that if you inhibit the focal adhesion with small molecule that inhibit the focal adhesion kinase, you will see a massive drop in release of insulin from pancreatic beta cells. And this goes through the paxillin pathway. So this is very nice. So we can characterize a lot of different things by looking at phosphorylation and sialylation at the same time. If you go further in pancreatic beta cells, now you know already a lot about them. Then there's also a lot of different model systems in mice to support work in diabetes type 2. And one of them is the DBDB mouse. The DBDB mouse has a mutation in the leptin receptor. And that means that this mouse over here cannot regulate its appetite. So it will eat and eat and eat, and it will grow and grow and grow. And that means that it will develop into a diabetic model at some point because of increased obesity. And if you take out pancreatic beta cells and look at the pancreatic beta cells between normal and DBDB, you will see that morphologically they change a lot. And this is supported here in this paper by Chris Rhodes, that is one of our collaborators. So if you look at mature granules, so you can easily see the difference between mature granules where you have the normal insulin packed, and immature granules where you have pre-insulin that doesn't work packed. So if you look at the DBDB, we have very, very small amount of mature granules, but we have very high amount of immature granules here. We can also look into Golgi and ER, and the size of the Golgi is massive when you look at the DBDB, and so is the ER. So there is a change in morphological towards larger Golgi, larger ER, larger cell type, and also immature granules. One of the specific feature with beta cell is that they are able to adapt again. So if you take these beta cells out and then you put them into glucose, very low amount of glucose for 12 hours, they actually come back to normal. So these are DB beta cells that are freshly isolated and then put into 12 hours of glucose stimulation. And here you have all the immature granulars and large ER and Golgi. And then it comes back to a lot of mature granulars and a lot of release of normal functional insulin. So this is called beta cell rest. And this is also suggested to be a treatment for type 2 diabetes and also proven actually. So we wanted to study this in a setup, and we have published this in 2 papers now here. So we have the mouse, freshly isolated islets, and then we have the normal mouse stimulated with glucose for 12 hours. And then we have the DB mouse beta cell and stimulated with glucose for 12 hours. And then we did the phosphor and sialylation study, and we used TMT or iTrack. And then we can see here, so this is the normal, and this is the normal plus glucose, and then it's the DB. And then we can see after beta cell rest, many of these changes in DB, they come back to almost normal. So the recovered phosphopeptides is more than 800 sites. So that means that these are very interesting for the beta cell rest phenomenon. And we can see here the regulated proteins, a lot of them are linked to the phenotype we saw before, like for example, ER and Golgi. So if we look at the thing we observed with the stimulation with glucose, we have the sialic acid and focal adhesion. And we can see here that this is very, very, very much changed in the DB version, but they come back to normal when you stimulate with glucose for 12 hours. And there are many of these that goes up and come back. This normally needs to more insulin release, but in our case, the beta cells from the DB, they don't have insulin, they have proinsulin to release. So that means that they release a lot of proinsulin that are not functional. So if we do KEC pathway analysis on the proteome, phosphoproteome, and sialylation, we can see a very big trend, very nice trend, that most of all these big KEC pathways that are involved are the ER processing and the stimulation or the insulin signaling taking place. And we have here focal adhesion, extracellular matrix, regulation of cytoskeleton. So all these together here show that there's something wrong in the secretory pathway. We can also go specific into enzymes that are involved in insulin processing. So we have the carboxypeptidase E and the converters 1 and 2. And we can see that they are going down markedly in the DB, which is the third one here. But we have also a lot of modifications that are going completely different ways here. And this is also the enzymes that are involved in the cleaving of the insulin, and we can see that they are also completely modulated. One of the other things when you look at the sialylation phosphorylation interaction is that many of the enzymes that are involved in sialylation, the sialyltransferases, here is shown 3 of them. Many of these are actually regulated by phosphorylation. And if we look down here, we can see that this sialyltransferase is actually the first one up here, the GAL1. And if you look at the sequence of the GAL1 and we go down here, we can see there is a disulfide bridge here normally. And this is made when this enzyme is not active. Then we have 2 phosphorylation sites that sit right next to, and these 2 phosphorylation sites, they go up significantly when we get the DB version. And what we also can see is that in the DB version, we have overall a lot of abundance in sialic acid compared to the normal one. But if you look at how you activate these enzymes, so this GAL1 here is activated by dimerization. And this dimerization is held together by 2 disulfide bonds that are made between 2 molecules, not the same molecule. In the inactive here, we have this disulfide bond. In the active, we have these 2 disulfide bonds between the 2 molecules. And the phosphorylation sites we see that are regulated are right next to this site. So we speculate that these phosphorylation sites are actually preventing this disulfide bond to be formed, so that we can form this one here that makes the enzyme active. And we can see this by increased sialylation. We also saw another enzyme. This is a kinase that we saw when going up with dB and down again after stimulation. And this is an enzyme or kinase that actually phosphorylate a lot of these targets here. And many of these targets are responsible for the modulation of the morphology of the ER and Golgi. So we speculate that this is a very central kinase here. And we can see it goes back to normal after low glucose stimulation. So in general, we can see by using proteomics and PTM analysis, we can see that all these morphological changes we have seen and observed, we can verify them by using our method. And we can see that after beta cell rest, they come back to normal. So this is very nice and very much reflected in both the phosphoproteomics and also the Sialylation study in this year. Another thing, just another example before I move on to the cysteine very shortly, is that sialic acid can be really, really dynamic. And this we have seen in nerve communication. So here we have the nerve terminal and the post terminal here. And we can purify these presynaptic terminals called synaptosomes, and we can actually stimulate them with KCl. And then all these small vesicles that are having a neurotransmitter, they move out to the active site and release their content and are taken back into the cell again in this vesicle here. We can purify the nerve terminal, and we can also purify the active zone or get an enriched active zone. And we have done this in this experiment in MCP in 2020, where we then looked at the D-glycopeptides in a similar way as before. And what we found was a substantial change after 5 seconds of depolarization. We see changes in all these proteins here that are sitting on the surface, either on the presynapse or on the postsynapse. So the postsynapse is not enzymatically active in here, but it's active on the surface in our synaptosome. And this is fully active because there is a mitochondria and ATP generation. So many of these changes here, they suggest very strongly that we have an enzymatic activity out at the surface that are putting on sialic acid, not only taking it off again. So when we purify these nerve terminals, there's no ER and Golgi present. So they have to come from other places. And the other thing is that there's no nucleus. So we don't know where the sialic acid is coming from, because it normally comes from in there. So there's a lot of big questions with this, but it's very, very nice to see that sialic acid is probably as dynamic as protein phosphorylation. The last part that I have left is cysteine modifications. So we have a lot of different ways that we can modify cysteines, and most of you know the disulfide bridges, but you can also have free cysteines, and you can have nitrous-related or sulfur-related. You can also have, if you have a lot of free radical stress, you can make cysteine into sulfonic acid or sulfinic acid, and then there's no way back. And then most of these proteins that go into here, they are either degraded or they can form plaques in many diseases. So many people have been looking at these modifications, and there's a lot of ways to enrich these cysteine modified peptides. So there are beads version, there are a lot of different ways to do it. And we also thought that we should jump in and make our own way to do it. So we developed in 2016 this CISPAT method, where we can mix these two compounds and make this compound here that look a bit like iodacetamide. So you can make a very nice modification of cysteines with a phosphonate group. And this phosphonate group here can then be used to purify using titanium dioxide. And yes, if you have seen this lately, this method has been reinvented in 2020 in Cell, almost in the same way. All these methods here are using differential reduction, so you can reduce with DCT, then you get most of it. You can reduce with ascorbic acid or other kind of things, and then you can get nitrosilation and all the other ones. So when we developed this, we thought it was a nice method, and we submitted to MCP, but they say, oh, you need to apply it to some biology. So what kind of biology did we have in the fridge at that time? We had Silac labeled control and HeLa control and EGF stimulated HeLa with a heavy. And so we agreed on doing this experiment, and we probably will not see anything with cysteine, we thought. So in this way here, we can digest into peptides, and then we can label with the cysteine with cispat, and then we can enrich both the phosphorylated one and the cysteine in the same time with titanium dioxide. And we did that, and very surprisingly, we saw that we had a lot of changes in cysteines. And then after we have resubmitted, we actually saw that the EGF signaling actually increase ROS production into the cell. So when you stimulate with EGF, you start a production of H2O2 in the cell, and that is able to modulate a lot of different cysteines inside the cell. So if you look into the cysteines that are, for example, here, the cysteines that are downregulated means that after stimulation, there are more free cysteines in this population. And we actually saw a lot of phosphorylation changes, which we expect to see with EGF stimulation, but we actually saw many more cysteine changes. And even on the EGF receptor here, we have all these cysteines here that are showing significant changes after stimulation for 5 minutes. And if you look more into the structure of EGF receptor, it looks a bit like this. So we have the kinase in here, and then we have the structure out here for the inactive EGF receptor. When EGF binds, it actually changes conformation. And during this conformation and change here, we have a modulation of cysteine bridges. And that means that here we have one kind of cysteine bridges, here we have another one. And here we can then stimulate, because now they go together as a dimer, and we activate the phosphorylation cascade down here. So there are many, many cysteines, if we look in this cascade, that are changing. So 5-minute changes, we see, for example, one of the more abundant changes we see here, where you get much more free cysteine, is in activated tyrosine phosphatases. So tyrosine phosphatases, they have this motif HC5XR, like this. And here the cysteine here is extremely important, because this is the active site. And then the active site of a tyrosine phosphatase, it needs to be free to conjugate to the phosphate group when you want to do dephosphorylation. And you can see here we have all these three. So here we have the PTPN1. And if you go into string, you can see that the closest interactive partner here is the EGF receptor. And the same here, the PTPRK, we have EGF receptor right here. So these are two phosphatases that specifically try to block the EGF signaling pathway after stimulation again. Other regulatory cysteines we find, the HDAC is a histone deacetylase, and the histone deacetylase have two cysteines. The HDAC 1 and 3, they have two cysteines here that are redox responsive. So that means that these two here control the activity of the HDAC 1 and 3. And that means that you can, by changing cysteines on the HDAC 1 and 3, you can change the activity of histone deacetylases. There's also a lot of zinc finger proteins that bind to DNA and other kind of things. And they are also relying on free cysteines here, because they are conjugating to zinc via the cysteines in this motif. And if you don't have the free cysteines, you cannot make this zinc finger, for example. There's also many of the redox-related molecules that you have in the cell, in the mitochondria, for example. They are also themselves regulated at the cysteine level after 5-minute stimulation. So we have all these ones here that are participating in all these conversion of cysteines, and they are all regulated at their own cysteines in this stimulation. If we look into potential crosstalking at cysteines, we can see, first of all, the tyrosine phosphatases are relying on cysteine oxidation. The acetylation is relying on cysteine. We also find OGT, which is responsible for O-gluconylation, that is also regulated on the cysteines. Netylation, ubiquitinylation is also, we find many of enzymes that are regulated on the cysteines, and also glycosylated related proteins or enzymes are regulated on the cysteines. So this shows that cysteine here has a lot to do with function of proteins. It has a lot to do with other PTMs and regulating enzymes that put on other PTMs. So this have brought us to the final, one of the final slides here is the PTM enrichment strategy that we are using currently in my group, where we have optimized it, and it's 2.0.042, because 42 is the answer to everything in the universe. So you can take cells or tissue or whatever, harvest the protein, and then you can label the free cysteine, for example, with cispads, then you can pull them out again. You can make it into peptides, multiplex them up to TMT 16-plex, for example. Then you can use this thiopropyl cephorose to fish out cysteine-containing peptides. So if you reduce in any way with ascorbic acid or DCT or TCP, you can fish out the cysteine-containing peptides. Unfortunately, the GE Healthcare is not making this cephorose anymore, so we have to look for other sources. Then you can do the titanium chromatography, and then we enrich for phosphopeptides, sialylated glycopeptides, and this cispad that we call SIA peptides. Then you can take from here, you can do CIMAC, which is separating multi from monophosphorylated peptides, or the second titanium. You get the sialylated or the D-glycopeptides out here, the phosphor and SIA peptides. You can pull out the phosphotyrosine by using antibody enrichment after you have done the initial pull down. We have done this and get not that many tyrosine phosphorylated peptides out, but enough. Then you can take the flow through from here and desalt it. And then we use antibodies to pull out the acetylated peptides. And then you can use other antibodies for ubiquitination or whatever you are interested in. And then at the end, we have the flow through where we have the non-modified peptides. And then we use these one for the proteome analysis. You can also complicate this a little bit more by separating membrane from soluble in the beginning here. Then you have 2 times as many fractions to run. But overall, this normally gives you between 80 and 100 LCMS fractions that you want to run on an LC system. So if you are paying for your LC-MS system, this can be a very expensive analysis here. But overall, this gives you a very, very high amount of knowledge that you can take with you for your signaling pathway analysis, for example. Okay, so we have a little bit of concern about these phosphoproteomics that are coming out now, and the reason why I put it in is that many people are using different kind of software. So many people think that it is very nice to see the site localization. So it's not only good enough to identify the phosphopeptide. You need to find the correct site of phosphorylation. And in many cases, you don't need that because you cannot upload it to pathway analysis softwares. But many people still do it, and that actually has been explored by many of the people that are making these softwares. So they have different kind of software programs where you can specifically look for the localization, and then you call them class 1 or class 2 or whatever class of phosphosites. So if you do this, you will see that almost every single peptide that contain multiple serine, threonine, and tyrosine will be identified with several peptide isoforms. And this is just an example from a data sheet I had the other day that you can see here. So this is the same peptide, different sites, same peptides, different sites right next to each other. And in many cases, the program cannot distinguish. So for example, in this case, it gives you all these 3 options, but it's maybe only be on one. So if you say that all these sites here are real, that means that you basically contradict the fact that kinases are consensus specific in most cases. So for the last 50 years, the research in kinase biology have shown that there are kinases that are very specific for these consensus sites. So you have, for example, R, X, X, S, or T, and you have S, T, P, and all these things. But in this case here, most of the programs that gives you all this abundance sites here will contradict this fact. So my view of this is that they're not real. In most cases, they are definitely not real. It comes back to the site localization, and you have to take into account what and think about what these programs are using. They are using the laws of water, and many of these have serine and threonine, and they are prone to lose water in MSMS. So many of them are probably error assigned because these peptides should lose water in any cases. So the conclusion is that analysis of multiple PTMs can increase the knowledge of signaling pathways. Comprehensive analysis require multiplex quantitation, so you don't want to do all these enrichments for a single peptide sample and then do it for the next again and do label-free. You don't want to do that. The order in which you perform the enrichment is actually important because you have to optimize the different buffer conditions. The methods we have developed have been used in many biological pathways: pancreatic beta cells, neurons, nerve terminals, T cells, and cancer cells, and it seems to work quite nice. When you do short-time simulation, if you do short time so that you go below 1 minute and you want to look at second stimulation, you have to have a mean to stop the stimulation again. And how is this done? This is extremely difficult. And you will see that almost the only thing that we have really been lucky with is either boiling SDS or ice cold 70% ethanol. And it's very, very difficult to stop the stimulation again. But if you want to have time points with low stimulation time, then you need to look into this and investigate your system for this. Okay, finally, I would like to acknowledge the people in my group that did some of the work. Our collaborators at AstraZeneca or MedImmune with Christopher Rhodes, and of course Giuseppe has been also co-developing many of these methods that I've shown, and of course the funding from the different sources in Denmark. Thank you very much.
Thanks a lot, Martin. It was a wonderful presentation. We had no doubts about that. So there is a slight delay between the line. So
sorry for that.
But no problem. We are actually, it's a pleasure that we have almost 70 people following. This is very good. So I mean, I would like to start some questions. Just waiting for the people to actually post some question. So Martin, this is mainly, let's say, a perspective in phosphorylation analysis, because now we know that I mean, the methods you have developed, they can really help us in identifying thousands, tens of thousands of phosphorylation sites. But the majority of them are on serine, threonine, and tyrosine. Actually, the vast majority of them. So my first question is, what is your perspective on these phosphorylations, like on histidine, arginine, glutamic acid?
So most of these phosphorylations have been found in bacteria. It might be they are in humans and in mammalians, but the methods that we've been using for phosphorylation studies have always been suspected to kick them off because of the pH values. However, I don't really believe so much in this, because I've seen studies where you take arginylated or arginine phosphorylation peptides and put it into acid, and it takes hours before they fall off. So I think maybe if people put it into the database search algorithm and try to search with this as an alternative, then you will probably see some. But you need to validate your data and see if it's really true that it's sitting there. I doubt that it will be a high stoichiometry modification if it will be there. But there are many people that are trying to look into means to purifying histidine and other kind of phosphorylated amino acids. But I've only seen it basically on bacteria.
Yeah. So histidine actually it seems to be a very hot topic, and as you mentioned in the beginning, there is, I mean, histidine kinases were discovered, I mean, many years ago. However, as you said in the beginning, the lack of enrichment methods for histidine, because it's acid labile. So the current method we have, they are not able. So I mean, maybe this is a message also would like to live in this forum is that, I mean, we need more methods for these PTMs, because the fact that we do not see many of them in even eukaryotes doesn't mean that they are not there. It's because we are not able to fish them efficiently. So
and then,
I would like
normally when you do the database searching afterwards, you never include it in your database searching. So it's difficult to know. And then in many of the peptides you identify, they have both histidine and serine and threonine. So
yes. So I have another question, but I would like to open for the question that were posted.
Can I make one quick one?
For sure, please.
Okay, so my dear friend, I have a question for you. First of all, I could see that like back in time when I was at Denmark, I remember, because then you said you showed us this 2.0 version, and I remember that was really hard for me to keep up to date in all the developments that you've been doing that time, because you're always like improving the conditions and so forth. So it's just a comment that how important it was, like as you were always striving to make the methods better. My question comes for the method because nowadays you said that you are pre-fractionating your phosphopeptides with high pH, but have you ever and assessed, because we know that some phosphorylations are basic labile, right? So in high pH they can be phosphorylated.
yeah, so when you say high pH, the pH is 9, and this is not enough for any beta elimination. And it's actually also, when we elute from titanium dioxide, we use pH 11,
Exactly.
and this is not either enough for beta elimination.
Okay.
So you need to have a much higher pH, and you need to heat it up, then you get the best beta elimination.
Okay, and my second question, and then we can go to the audience, is regarding the phosphatases, because it's, as you showed, they're like highly abundant. At least in my hands, it's not always that we can get some phosphatases. Like, for example, it's difficult to find them, especially phosphorylated. So do you think it's due to time that we are looking for? Because you said that most of the time activated really fast. So do you think?
So you mean the phosphatases, how the phosphatases are activated?
Because usually, at least in my hands, when I have the data sets, sometimes we can get kinases regulated, but it's really hard to figure out the phosphatases in our set. So do you think?
So I don't really think that phosphatases are regulated by phosphorylation. I think they're regulated by other things. So in a recent study we did on T cell signaling, we find regulation on the sialylation on some tyrosine phosphatases after stimulation, short time stimulation. So I don't think that it's that these are regulated by phosphorylation, actually.
Okay.
But they're very important that you kill them with your method of stopping your signaling. So you can add there are phosphatase inhibitors a lot, but they don't only work to a certain extent. So the best thing is to denaturate everything as fast as possible.
Great. So Martin, we have many people from Brazil, but also from abroad. Simone, very nice to see your here. So, but I will, and many people actually congratulate you for your presentation. But you will see that Thiago will post. So the first question is from Magno. He said, during the cell cycle, the cells have an hyperphosphorylation state, and most phosphorylation have no biological meaning. How can we fish or validate the good ones?
Well, you just have to do a lottery or something like that, or you can maybe employ 1,000 PhD students to start working on it. But there are examples of things that you haven't. So the problem is that very few phosphorylation sites know what they're doing. So, but I believe if you see when you do, for example, stimulation, and you see a change in a phosphorylation site, that change will be significant for the biologic background or the biology. So there are many that doesn't change whatsoever you're doing with the cell. And these are what people are called background phosphorylation. And they can be, because sometimes these kinases could be promiscuous, and they can, maybe if they get over-activated, they will phosphorylate by accident. But no one really knows. And there's also the problem with the stoichiometry. You never know how much of a single protein that is phosphorylated at a given time. So it could be that a very abundant protein gets phosphorylated with 0.5%, and you still see it as very abundant. So I don't know if that was the answer for the question, but I think that it's difficult to say that a phosphorylation site is not important.
Yes. Yeah, I think it's very difficult to find the right answer to that question.
Yeah.
Anyway, so also Marcella said great talk, and she would have liked to see you on video, but we have to say that we had some little issues before, but we managed.
I look great. I look great.
Great. So now from the Laboratory of Neuroproteomics, Danielle is asking, thanks Martin, I wonder if you can comment on functional experiments, not MS related, for post-translational modification validation.
Yeah, so this is one of the big problem in PTM omics, is the functional aspect. So you can do a lot of different things for investigating phosphorylation sites. For example, you can mutate them. You can do peptide pull down or protein pull down to see what binds specifically to the phosphorylation site. We have been speculating a little bit in if you have a nice biological setup where you have a readout, for example. So we are stimulating nerve terminals or neurons, and we see release of neurotransmitter. And if you want to see if a protein phosphorylation is involved in such a biological process, and you can measure the biological process, you can actually make synthetic peptide with a pentratin tag. So the pentratin tag is a small tag of amino acids that actually drive the peptide through the plasma membrane and into the cell. And if you add enough of this to the cell, then you will have a huge amount of phosphopeptide or diphosphopeptide with the same sequence inside the cell. And when you then stimulate, because many of these interactions are taking place in a linear interaction, then you can outcompete the interaction between the real proteins inside the cell. And that will affect the outcome of your biology. So for example, if I have a protein where I have a nice increase in phosphorylation on a protein that I know is not driven by, where the phosphorylation is not driving activation, but I suspect that it is driving interaction, and that interaction has a lot to do with release of neurotransmitter. If I add excess of this peptide, then I will see that the neurotransmitter release will be driving down, so that I see less neurotransmitter release, and then I can say that this specific amino acid or phosphorylation is involved in exocytosis, for example. But it's very difficult to study these things.
And this is for phosphorylation. Imagine for
this is phosphorylation. If you go to sialylation, it's a nightmare. So glycosylation is very difficult to study the function on. So you cannot remove site-specific sialylation on a glycan structure. You can knock down cellular transferases, but that will affect many targets. You can remove the site in the protein that has the end-link glycosylation, for example, but then you have the risk that it is not folding up correctly, and if it's not folding up correctly, then it will be sent to degradation after the ER. So that is very, 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 very difficult to look at.
Great. So as I was saying, there are many students that say great talk, Gilberto, Carolina, but also Gilberto Domon, your friend. Yes. Dear friend, thanks for enlightening us so much. This is
Well, there's no light here in Denmark. It's getting dark now.
Can I make a comment on that? I've been told that today is Gilberto's birthday.
Congratulations.
So,
happy birthday,
Gilberto.
I thought it was in December.
Happy birthday.
So, Martin, the next question is from Reinaldo Melo, and he wants to know, how do you normalize the abundance of phosphopeptides for the protein abundance?
Yeah, I think so. This is one of the reasons why I really love short-time stimulation, because in short-time stimulation you don't take into account changes in protein level. And there has been some papers out. Stephen Gigi published a paper, How to Normalize. And I think most of the attempt to normalize is that you take the ratio and then you normalize ratio-wise. So if you have a protein that change by 1.3 fold, and you have a phosphorylation size that change by 1.6, then you have an increase in phosphorylation. But in this scenario, there's no taking into account of standard deviations. And this is the big question. So can you just subtract ratios? And I don't believe that you can do that. So I actually suggest that you don't normalize, that you just treat them individually, and then at the end, when you have looked into the biology, you can start to say, okay, I'm very interested in this phosphorylation. Now I go and look at the protein. So it requires much more work, but I think this is the only way to go. I mean, unless someone comes up with a nice algorithm that takes into account the standard deviation on protein and peptide level.
Which is different, of course, for at peptide level compared to protein. But then you merge several peptides. Yeah. So another question from Magno is, I'm curious about how the treatment with low glucose stimulation works for type 2 diabetes.
So it's so I think it's similar to the many diabetic people. They go on diet. The moment you go on diet, you actually see a change in the insulin secretion in type 2 diabetes patients. But of course, it's not something that so for example, if you take the beta cells from DB mice and you treat them for many days with this, then you see that they come back to normal DB. So it's not a DB mice they're not human. It's only a phenotype that reflects diabetes, obesity-induced diabetes. So people are looking into this at the clinic, and I think that it will be more reflected in these diet programs. So you will see it with the diet programs.
Yes. So there is a Fabio Nogueira, also a friend, and you know him. So Fabio say, how do you determine the subcellular localization of phosphoproteins, focusing on the one that you don't? Okay.
Well, it's difficult because you're homogenizing the whole cell. So for this, you need to have a cell. You need to have compartment enrichment. We have tried to do a little bit with this with other people here, but it's very difficult.
If I can add something to that, Martin, I remember that there was a cytochrome C phosphorylation at some point that most of the people, they missed. And the reason why they missed is because when they were trying to isolate mitochondria, they were not adding phosphatase, because normally you start with bova and heart, so you need liters of phosphatase. So actually this comes back to what you said in the beginning of your presentation, to take care about the phosphatase activity, because if you don't inhibit, then you will. So just to add something to your question, maybe when we think about subcellular localization and also fractionation, different compartments, if you want to study phosphorylation, maybe you should think about adding phosphatase since the beginning of your protocol.
Yeah.
And Fabio also asked, how do you do site assignment and motive prediction in the phosphoproteomics, I guess, which software you use?
So we are mainly using PD, Proteome Discoverer, and that has this built-in phosphor-RS localization. And we don't go below 90%, so we go higher if we can, so that we are confident. But you have to be aware that there are not many programs where you can actually use the knowledge about the site. So when you do the phosphopeptide identification, then the peptide is the identification mean. So that means that if you identify a peptide with a phosphate group on, this is with 1% false discovery rate. So that means that this peptide is present, but maybe you don't know where the site is. So don't ever throw that away, because you can only use the site for very few programs. So for example, if you want to predict what kinase is sitting there, then you need to know the site, of course. So you take the one that you can assign well, and then you make this motive X, and then you can predict what kinases you might have there. But also be careful here, because there are 537 kinases, and we don't know the consensus site for more than 150. And if you look at many of the new publications where they are taking everything, isoforms, then it seems like there's no consensus site. So it kind of contradicts each other, that you want to do that, but still you want to take everything, and not only the most confident one. So so we are using Proteome Discoverer, but you can use all kind of other things for this.
So Martin, if I can follow up on this comment, because I mean you showed this slide with this table with the phosphopeptides. And at the same, this was a very important message, but at the same time very scary, because basically what you were saying, that the majority of this phosphopeptide that we identify, although we use a phosphosite assignment localization, which is above 90%, they might be wrong, because most of this site assignment localization are based on the loss of water, so the presence of the hydroalanine, and this can happen in the gas phase for other phenomena. So what is your solution to that?
Well, I think if you can build in that you also look at retention time, because there are some cases where you can very clearly see that you have two species from the same peptide, but in many cases, if they are just some amino acid from each other, they actually separate in the reverse phase separation. So you should build in some kind of means that ensure that the two isoforms are away from each other in the separation. Then you will be able to deal with it. And it's also strange when I look into my data. If I look into my isoforms data and I see the same peptide with one phosphor group but several places, they are almost all regulated in the same way. And if there was different kinases that worked on this area, you will expect that after stimulation or something like that, that at least one of the isoforms will change in quantitation. But it's very rare that you see it.
So, So do you think we should go back, or at least use more the ETD fragmentation? Because then, I mean, this problem will be less.
No, I think that you should not spend so much time looking at the site and not be so focused on the site, because if you want to do pathway programs anyway, you cannot upload the site. So, and there's not many sites where you know what it's doing anyway. So the best thing is to do it on the protein level in the beginning, and then if you're really interested in the biology of this area, then you go in and manually find out what the site is. Because when you do manually, you will be able to see the intensity of the peaks in the MSMS spectrum, and then from that you can very often see that, okay, it's more likely that it's this one or this one.
Okay. So what you basically say to us today is that, I mean, these tens of thousands of phosphorylation sites not all of them might be correct, although if we use stringent criteria for site localization. And we should go back.
So what I basically do now, now I look at peptide groups. I don't look so much at peptide isoforms. So, for example, I have a study with the active zone of nerve terminals, and I find 18,000 unique peptide groups, phosphorylated peptide groups. But if I open up and allow for all the possibilities that I get from PD in the isoforms, I have 28,000. So, and that is a big step, but most of the one that I have double with different sites on, they are regulated exactly the same way.
And the retention time is the same also, maybe.
Yeah, more or less the same, yeah.
Less the same.
Yeah.
All right. So I'm taking time from the audience. So Marcella, she said, thank you very, very much for your exciting lecture. She's a very big fan of you. We know that.
Don't tell Tiago.
Her question is regarding the sialic acid. So we move now to the glycosylation. So there is a huge discussion on UGC as to be immunogenic to humans. And she asked you, do you think we do not produce the new GC?
Nope.
Because this could generate, when we have it, an autoimmune disease in some cases.
Mm-hmm. Yeah, so we don't. So we have the enzyme, but it lacks these 92 base pair deletion, so it's not functional. So so there's no way we can actually make it, but we can take it up from the surroundings. So this is also probably why you see it on many cancer surfaces, because they grow fast, so they need nutrition from everywhere. So they will just take it up, and many of these cellular transferases, they can actually incorporate it into proteins, I think.
Yeah, I think there are even antibodies against some of these sugars that are using cancer to recognize cancer cells.
Yes.
All right. Another question from Marcella. Is the best normalization would be to have the same peptide identified, unmodified, and modified? Yeah.
No, not really, because you in the case of the modification, you enrich. So then it comes back to the stoichiometry. So if they were all 100% stoichiometry or so, then you will see it. So when you do a lot of TMT and ITREC, you have many times you have a co-isolation, and then you don't see the correct change. So that means that if it's not a really big change, so you can see a big change if you enrich a phosphopeptides, but the phosphopeptide is only there in a stoichiometry 1 to 100. Then you will see, and you see a change in the enriched one, but then you will not necessarily see anything in the non-enriched. So imagine you have a phosphopeptide and it changed by 50%, but it's only 1% that are phosphorylated. Then you see a very big change in your phosphor enrichment, but in your non-phosphor you don't see it because it's going below your standard deviation.
And I mean, yeah, this comes back to the question of the stoichiometry, right?
Yeah.
And I mean, there are sites that change a bit, but also there are sites which actually have high stoichiometry, and especially
this one could be okay.
Yeah. All right. Just coming back to the version 4, what did you say? Version 2.0042. Actually, Gilberto posted a message that he said that he is at version 8.6, which I guess are the years.
Yeah.
So happy.
But you know, 42 is the answer to everything.
All right. Thiago, do you have other questions?
No, I don't. Just want to congratulate again and thank you very much for accepting to give the talk.
Welcome, my dear friend.
My dear friend.
Thanks a lot, Martin, for your wonderful presentation. I think
you are welcome.
Everyone here liked a lot. So
yes,
I hope to see you in person soon.
Yes, Okay,
You too.
thank you.
Thank you.
Bye.
Bye. See you guys.
See you.